Hello guys, welcome to Learn Extra Live. This is Life Science with Aslam. How are you, Aslam?
Katlego, I'm great, and yourself?
That's great. So what are we doing today in Life Science?
Let's just do nothing today.
Let's just relax.
Let's just look at each other,
Let's just do that.
and we look at them, and we take it from there.
And there you go. I think that's perfect.
Now
But I think
today
people
we are
fight.
we are pressed for time. We need to do some revision on paper 1 items, especially on the first half. We're not touching evolution at
okay.
all. We're going through some things on genetics, some things on meiosis,
Okay.
and other stuff as well.
That's very important stuff. Thank you, Aslam. So guys, of course, such an important show for you grade 12s because it forms such a big part of your prelims and your finals and everything that you need for next year for university or just to pass matric. So make sure that today you are so alert and alive because it is so great to have you guys here as well from me. So join me on Facebook as well, facebook.com/learnextra, as well as on Twitter. And our Twitter handle is @learnextra. This is where you can write anything that you wish to, guys, especially questions on what you're not understanding or what's been giving you problems, or maybe something that you got wrong in prelims that's been bugging you in paper 1. Post that to us so that we can help you. Also, another important thing, because Learn Extra Live is always here for you and Mindset is always here to catch you, we are having a Saturday show where we go through the prelim papers. So make sure that you can start posting your questions now on the page so that on Saturday there's no worries. You can just maybe repost or resend them on Saturday. So don't forget to join us for that, guys, because it'll be so important. And the schedule will be up soon for what subjects will be covered. So keep your eyes posted on the page. Over to you, Aslam.
Thank you, Katlego. Welcome, guys. We are at that stage now. Your holidays are over. I can't even call it a holiday. Your last break is over. We are now on that steam train that's moving fast ahead. So you better be on the ball. Keep yourself healthy. Make sure you get enough sleep, not too much, enough sleep, and you have enough time to study everything. Do not make the fatal mistake of learning from now till the 22nd of October for your first paper. And don't learn anything else. And then from the 22nd of October, you learn every day for the next paper. That is a fatal mistake that many matric learners, or all learners do. They check their timetable, they prepare only for the first paper from now till then, and thereafter they're giving 1 day per paper. That's not the way to go, guys. You'll be in serious problem. We're looking at paper 1 items today, and since we haven't touched on comprehension type of questions in the last couple of shows, we're looking at one, and this one here is taken from the Eastern Cape Prep 2012. Yes, guys, from 2012. So you had your paper in your province, wherever you are, and this one is from the Eastern Cape. They say, read the article below and answer the questions that follow. Obviously, that is the instruction that they're giving you. You've got to read the article. And it's not a very short article, it's quite a long article, so you need to read it carefully. They say business development, SA produces more GM crops. Again, there's the connection to your syllabus. We're talking about GM, and by the way, for those of you who still don't understand, GM is talking about genetically modified. So GMO, genetically modified organisms. So this one here was 28 February 2008 by Michael Apple. Since the adoption of biotech crops in 1996, the amount of genetically modified GM maize, soya, beans, and cotton planted in South Africa in terms of area planted has increased to 1.8 million hectares. Wow, that's an amazing figure that they're giving us, has increased to a staggering 1.8 million hectares. White GM maize totaled 1.04 million hectares, an increase of 48% over 2006-2007. In 1 year, or 2 years, this is 2008, 2006 to 2007, over and above that, 48%, representing a market share of 62%. Yellow maize increased from 528,000 hectares to 567,000 hectares, up 7%. Agri SA President Luri Bosman said on Wednesday. That's what he said. GM crops are engineered to withstand environmental factors such as drought. Certain seeds have a dominant trait of herbicide tolerance, allowing farmers to make use of herbicides without harming their crops. Notice what I'm doing as I'm going along. I am underlining the key words. AgriSA is positive about genetically modified agricultural crops and is enthusiastic about the contribution they can make in the future towards increased production. Bosman said. The most popular GM trait in South Africa was BT insect-resistant maize, with 1.1 million hectares planted, followed by RR herbicide-tolerant maize, with 373,000 hectares planted. Stacked trays BT plus RR sales commenced in 2007, and 80,000 hectares have been planted to date. So you can see there's an increase all the time. Field trials of drought-resistant maize varieties, a trait that has since been incorporated into other crops as well. Of what again, by the way? Drought-resistant maize, are already underway in South Africa, and the technology is expected to be commercially viable by 2011. That's last year already. So a big, long article that you had to read for this. And again, guys, the language issue comes to the fore here. If you cannot read properly, you cannot read a little fast, you are going to experience problems. But the key words you underline, read it as slow as you want to, not too slow that you have to take the whole 2.5 hours to read this. That's not going to be good. But read it carefully, underline the keywords as we've been doing here, and then you go straight into the questions. According to the data provided in the article, predict the future trend regarding the use of modified crops on South African farms. Obviously, you saw what was happening in 1996, 2006, 2007, 2008, and then a projection for 2011. So the answer there obviously is that there would be an increase in the use of GM crops. The key word there being an increase. And how did we pick this up? We have seen from the data provided from 1996 to 2006 there was an increase. From 2006-07 to 2008 there was a further increase. And they're saying that by 2011 there would be more technology available for this to even further increase. Okay? With reference to the article, mention 4 beneficial traits that could be incorporated into the crops to raise the competitive edge of the South African agricultural sector. The bottom line here is up to here. With reference to the article firstly. It must be from the article, not from your head, not from the textbook, from the article. Mention 4 beneficial traits. The rest is obvious. You can put in brackets even to understand that. Now, obviously, we need to go back to the article. Beneficial, and we've been marking them as we went along. The first one there is to withstand drought. So they become drought resistant. Then they said further that they become herbicide resistant. So one is they become drought resistant, and the second one is herbicide resistant. Now, let's not confuse that. Herbicide is not an organism, because some of you may read an article like this and think herbicide is an organism. Herbicide, like pesticide, is a chemical that will damage or rather kill weeds, herbs. Herbicide, pesticide. So pesticide is an all-encompassing term for any pest. Herbicide is specifically for plant weeds. So what happens normally when farmers spray their crops with herbicides, the herbicide can damage the actual crop that they're trying to save. So here we're talking about herbicide resistance. So one was drought resistant, the other one was herbicide resistant. The next one here was insect resistant maize. And if you look at throughout the article, they spoke about an increase of the plant. So if they want 4 benefits, then we would say one was drought resistant, 2 herbicide resistant, 3 insect resistant, and 4 a better yield. A better yield means we get more crops. And this obviously would give us the competitive edge that we need as a country in terms of the agricultural sector. Name 3 genetically modified crops mentioned in the article that are being planted in South Africa. Guys, that was said in the first sentence already, if I remember. There we go. Maize, soya, beans, cotton. There's 4 of them there. We needed to mention 3. Let's go back. Name 3 genetically modified. So it is maize, 2 soya, 3 cotton, and 4 beans. Again, you did not need to memorize anything for this answer, guys. This answer came from the article. So whatever we've answered so far, that's 3 marks there. 4, sorry, let's go down again. That's, the first one was 1, and if you go further down, it's 4, it's 5, and this 3 here gives you 8 marks already, just from the article. Give a reason why high priority is given to the development of drought-resistant crops in South Africa. And that is a general knowledge question in the sense that we know our country and we know the problems. And the first problem is that we do not have very high rainfall. And we do not have, let me write that, do not have many rivers to irrigate from. And remember, when we're talking about this, we're talking about rivers that will reach all the rural areas where this is taking place. We may have rivers, yes, but they do not reach all the rural areas where we need them to reach as well. So that's our reason there. We get 2 marks there. They say, mention any 4 objections. What happened there? Mention any 4 objections against the introduction of genetically modified crops. Now, guys, the last 2 questions, the one before this and this one here, are now going to your knowledge and not from the article. They want to know 4 objections against the introduction of genetically modified crops. The first one would be, we do not know the long-term effects. And remember, they asked you here to mention. If you need to explain, then you'll have to talk about that there may be long-term effects, in other words, health, etc., to this. Number 2, this genetically modified may interfere with nature. In other words, with the ecosystem. it may cause an imbalance in the ecosystem. Some organisms may have advantage over other organisms, and they may outcompete them, and because of that, they will survive, others won't, and we'll have a problem with that as well. 3, we don't know whether we may introduce some diseases with this as well. There's big debates going on at the moment throughout the net, throughout the media, where you get those that are in favor of genetically modified organisms and those that are against genetically modified. And one of the biggest arguments against genetic modification is that these organisms that we eat or the plants that we eat may cause problems for us health-wise. So they may introduce some diseases, or you can even say side effects. 4, in terms of genetically modified organisms, we may be playing, so this is a religious objection. We may be playing God and interfering with the natural things. Remember, here we spoke about interfering with nature in the terms of ecosystem. Here we are talking on terms of religion. And there are a host of other objections you may also use in terms of this. For example, it may be expensive. It may be expensive, and not everybody may be able to afford it. And it can also create monopolies, because the companies that manufacture these will make sure that these organisms do not reproduce naturally, and you have to buy the seeds. So you can create monopolies that way. So in that way, we're not alleviating poverty.
Aslam, before you carry on, can we take a quick break?
Let's go to that break.
Let's do that break, guys. During this break, maybe just get a glass of water or get some tea, but make sure that you join us as soon as possible, because it does get better from here. You know that. See you right after this short break. Welcome back, guys. If you have just joined us, which I hope you haven't, I hope you've been here from the beginning, but we are doing revision and going through paper 1 on life science. So make sure that your pens and papers are out and you're writing things down and maybe making notes for yourself where you're like, oh no, that part gives me a bit of a problem. Put a bit of a star next to it, and maybe even have a piece of paper next to you where you write down questions that have been giving you problems, because there will be another show on Saturday that'll be going through prelim papers. So make sure that you have all your questions up to scratch, so that when it's time, you just shoot them through. But for now, you can still ask questions on what Aslam is doing, because we're here to answer every question that you guys need. And if you're perhaps confused or anything, just write it on the page and I will get back to you, I promise you. Over to you, Aslam.
Thanks, Katlego. Guys, thanks for your questions coming through, and for many of you who have asked about the disadvantages or the arguments against GM crops, and we have discussed that in the last question. And like I said, we could add to this list as well. For example, adding pest-induced problems into the crop as well, and that can cause other problems along the food chain as well. And if something goes wrong with the genetically modified plant, if it is then taken into the food chain, then it affects every organism along that line as well. The next question says, and I like the way this question is asked. Normally, they would say give 4 arguments against and 4 arguments for, or give advantages and disadvantages. The first question said, for objections, that was straightforward. Now, what's this question, how it's worded? If you're a fruit and vegetable distributor, which three trays would you like to be incorporated in the agricultural products? A wonderful way to ask what are the arguments for genetically modified organisms, or what are the advantages. So they didn't ask you for the advantage, so you've got to read carefully, guys. So he's asking you, why would you do it? And the only reason you would do it is for the advantage. So that's what it's asking you here. And what do we say here? One is, we say very clearly that we would want drought resistant fruit and vegetable. That's the first thing. We've already discussed it earlier. Two, we would like larger let me just write that there, something wrong with the pen here. Katlego, we're having problems with our pen again.
Oh, okay. That's perfect.
Larger fruit. We would also want fruit that have a longer shelf life. Okay? So those are some of the reasons we can give there. We also want a larger yield. Why would we want a larger yield? Because the yield means how much of crop we're going to get. The bigger the crop we're going to get, the more we can sell and the more money we would be able to make in that way. So we're looking at the arguments for this type of thing as well. So it would give us drought resistance, and also you can put drought, insect and pesticide resistant fruit. Larger fruit there, we got that there. Let's just take that away. Oops. Larger fruit and longer shelf life and a larger yield. Last question in this question, or the last sub-question, what public concern could be addressed by labeling all GM foods in our supermarkets? This is a very important and contentious issue. All people that are nature lovers or natural Greenpeace organizations and the rest of them, they insist that all supermarkets, and in many countries in the world, it is law for the manufacturers and the wholesalers and the retailers to put on their packaging that this particular item has been genetically modified. And what public concern could be addressed? Obviously, the right to information. And interestingly, this also is enshrined in our Constitution, the Access to Information Bill. That means you and I all, we have the right to access to information, to get information about certain things. And this is something very important for us. We are eating, we are consuming this particular material. So therefore, we have the right to get information if we label that there. The right to know. In other words, we have the right to know. That's all we're saying there. Let's move on. Katlego, that other learner that asked us a question about recombinant.
Yes.
Good.
Munaka asked about recombinant
Her name
DNA.
again?
Munaka.
Munaka,
Yes.
well, this question here is going to address what you asked. That's why we haven't answered it separately. So pay attention, my darling, because this is going to help you to understand it. I love the types of question here because it encompasses this section in a nice way where you can understand it, and at the same time, you can see how they twist and turn with the questions. The diagram below shows a modern technique to manufacture insulin. Study the diagram and answer the following questions. There they have Bacterium E. coli, and they have a little circle there labeled A, and you see something with that circle moving that way, and enzyme B is used there. That comes down, and then we're taking a gene, human cell with insulin gene. We're taking that gene, and you can see it's being inserted there. using enzyme C, and you're going down there, and you're coming down this way, and we're taking this particular organism again, and we're having that whole story there going on there. A whole lot of things happening there, and then eventually we have insulin manufactured for us. So important, guys, to go through the diagram to understand what the diagram is telling us. Don't take it for granted that you go straight to the questions. You first go through the diagram properly. See what is happening there. And then you go to the questions. Name the technique used in the manufacturing of insulin. Oops, what happened there? What is this technique used in manufacturing insulin? This is the very technique that you spoke about. Recombinant. Oh, my pen is giving me hassles. Or we can say the word to say biotechnology. Identify part labeled A. The circle inside the bacterium E. coli. And guys, if you know the section, then obviously you would be able to label A as, and I do it on the diagram for you, that would be the plasmid. That means, and what is the plasmid, by the way? The ring of DNA. The ring of DNA. Bacteria is made of a ring of DNA. Remember, bacteria is a prokaryotic organism. It doesn't have a true nucleus and other organelles. So therefore, it would not have a nuclear membrane and a nucleolus and so on. It simply has nuclear material. So we take this thing out there and we move on. Let's move on to the questions. So we said that was a plasmid. Supply the names of B and C, enzymes B and C. B is called the restriction enzyme. Oh God, the pen is giving me nightmares.
Is it giving you problems today?
The restriction enzyme would be B. That would be answer to B. And C, this one here. Now watch the two enzymes. The first enzyme is used to cut this plasmid, to make a space in it. It's like a chemical scissor. This is called restriction enzyme. And the second enzyme is used to insert a gene into this gap that we have. So the one that we're cutting with is called a restriction. Please don't get me wrong. When we say cutting here, we do not mean cutting like with a scissor, but we are talking about chemically cutting something. And enzyme C that is coming in there is known as ligase. Ligase, okay? So we've given the names of those two. The next one there, what type of cell division takes place at D? That's a very simple question, guys. And notice as we're going with this question, the question is actually developing this whole section on how this process unfolds. What did we do first? What is this thing? And how did we cut it? How did we put something in there? And now we're saying, what type of cell division takes place at D? D is this area. And you can see what's happening here, that this cell is becoming many of the same, identical. So that is only one type of cell division, guys, and that is mitosis. Not yourtosis, mitosis, okay? And remember the difference between mitosis and meiosis. Mitosis, exact duplication. Meiosis, reduction, division, where the cells that are formed are different. Still moving with that question. Mention any two advantages of the above technique. Now here we've got to pause for just 1 second. Please understand that this technique was used after in the old where they used to use insulin from animals. So when we look at the advantages of this technique, we must look at the advantage of this technique as opposed to the old. And there we talk about, first of all, the fact that there are no side effects. In other words, when we use animal insulin, there could have been side effects, allergies, etc., that could have affected us. Now we are using human insulin, so obviously there shouldn't be any side effects. We can produce large quantities in a short time. There would be no religious objections. Remember, there are certain people that cannot use certain parts of certain animals, so they would have problem using animal insulin, etc. So this way here we're using humans insulin, so it won't be a problem. And we can also say that it would avoid killing animals. Now, this question I love. It says, name another unicellular organism that can be used in the above process. Now, I bet many of you would not know the answer to this one because it would not generally appear in a textbook or in your guides, etc. We only talk about bacteria normally. But you needed to go back to your grade 11 work to think about this one. And the unicellular organism that we use in the above we could use is yeast, believe it or not. And they want to know, give one reason for the unicellular organism mentioned. Why do you choose yeast? Obviously, yeast is easy to find, and it is easy to cultivate. In other words, they reproduce fast, and also it is cheaper than using bacteria. That, guys, deals with this important process of recombinant DNA and how we work it out. Now, please just figure it out quickly again. The bacteria is taken, we take the DNA out, we cut a portion of the DNA, we take a part of insulin from the human, we put it into that particular plasmid, and we reproduce that stuff. The diagram gave you the whole process. The next question we're going to be looking at shortly is the diagram below represents certain stages of a meiotic division in a certain plant. Study the diagram and answer the questions that follow. So here's the diagram. I'm going to show you the diagram quickly, and then we'll go into the questions immediately after. We'll give you a short break to work it out. So let's look at the diagram quickly. Here's metaphase 1. They're telling you that this is metaphase 1. Here they're telling you this is anaphase 1. And you can see what is happening there and what is happening there. And from there, we're getting something there, diploid ovum, and we're getting a haploid sperm, and we're getting a zygote that is forming there. Guys, that is the diagram that we need to look at. So with that, let's stop there, pause, think about that diagram. Let's go for a break. We come back, we go to the questions.
Okay, awesome. So you guys heard that. I hope you've got it down so that you can start answering it during the break. I know you guys are smart, so you'll probably send through your answers before the break even finishes, because some of you say, it's such a long break, so I know you know the answer already. Post it on the page. See you right after the break. Welcome back, Mindsetters. Of course, I know that you guys are enjoying the show because it is such an awesome show, and you guys are already noting the importance of it. It's so important, guys, that you've joined us today because it helps you so much for your finals. Like, Now, unfortunately or fortunately, there's no excuse not to do your best. There's no excuse not to do well if it is your best. Because if it's your best, it won't be below average. Trust me, your best is over 100%. So give it your all, and I promise you the results will follow. And of course, you guys know that we are way past the 19,000 border on our likes. In fact, we have about 15 more likes to go until we reach 20,000 likes on the page. If you haven't liked the page and you're sitting in your seat right now, please jump over on your phone, on your laptops, on your computers, on your anything at school, with your mom's phone, your dad's phone, anything. Just press that like button, guys, because 20,000, wow, that's a big one, and we've worked so hard to reach this point, and I'm ready to celebrate that 20,000. Over to you, Aslam.
Thanks, Katlego. If you can't like the page, how can you not like Katlego, guys?
Exactly.
So then you should press it about 60 times just to like Katlego. Right? Guys, one question was from
Let me show you
one of
who
the
is it
learners
from.
there, wanted to know a clarity on
Nathan.
3.2.4. The name?
Nathan.
Nathan wanted to know why the answer to 324 was not binary fission. Let's go back to the question. The question says, what type of cell division takes place there, Nathan? They did not ask what process takes place. Binary fission is a process, that is true, but here they ask specifically, and see how important it is to read the question. You needed to answer the question in terms of a cell division. There are only two types of cell division, that is mitosis and meiosis. And in this case, it is mitosis. Binary fission is the type of reproduction that bacteria undergo. So it's a process. And how does binary fission take place? It takes place by cell division called mitosis. Good. I'm hoping that clears your question, Nathan. So we were with the meiosis question. So we looked at metaphase 1, we looked at anaphase 1. We find that there's a diploid ovum, and what's happening on this side, we have a haploid sperm, and the zygote has formed there, A. The question says, mention an observable characteristic feature of metaphase 1. Observable. What can you see? Very simple. Metaphase, meta in the middle. I must apologize for my handwriting earlier. Lots of you are complaining about the handwriting earlier. I think we fixed the problem now, and you'll bear with me, and you can score my handwriting now from now till the end, and I'm sure you'll give me 11 out of 10. Okay? Katlego, you think I'll
Let's
manage?
hope so, yeah.
I hope so. We're getting too spoiled with the typing and too little writing. That's
Yes,
the problem.
that's the problem.
Okay, so they want to know an observable reason. So first of all, look at the cell. And in this particular cell, these are the poles. The top and the bottom are the poles. In another cell, we could turn this around. We could rotate it so that the poles are on that side. Okay, so you must understand that the cell can go in two directions in this case here. And in this case, you can see that's a pole, there's the middle, the chromosomes are in the middle, but because it's meiosis, we can't just say that the chromosomes are in the middle, we say that the homologous chromosomes are at the equator. Homologous chromosomes are the equator. This is very important because this is division 1. If we simply say chromosomes are the equator, it could be either division 1 or division 2, and that's dangerous. So we rather say homologous chromosome. 3.3.2, name the process that takes place in prophase 1 that leads to variation. So in prophase 1, there's lots of processes taking place. They're not interested in all the processes. They're only talking about that process that brings about a variation in the offspring. And, yes, you guessed right, that is crossing over. By the way, some questions like to ask, what is the significance of crossing over? Or they show you the crossing over in a diagram and they ask you, what's the significance of that process? And then the answer to that would be crossing over. And remember, there's not the answer to this question. The answer to this question is what I'm underlining. If we ask for the significance, then we say that crossing over allows for exchange of genetic material which in turn brings about variation. What do we mean by that? We mean that genes from one homologous chromosome is swapped with another homologous chromosome, and in this way we have the story that, or rather one homologue of the pair. One chromosome of the homologous pair. Let me rephrase that. One chromosome, a gene from one chromosome of the homologous pair, switches with the other chromosome of the homologous pair. This is crossing over. So the genes have swapped places. So the chromosome that was there is not the same as it was before crossing over. And this brings about variation. This is one of the most important ways in which variation is brought about. And this is one of the most important significances of meiosis, to bring about variation, that despite Katlego and I both are descendants of Adam and Eve, but we look so different. She is so beautiful, and I am the ogre.
No, not really.
As long as you can say that, then I'm okay. Then I'm happy. You've made my day, Katlego.
Thank you. I'm
Thank you
glad.
so much. What error occurred in anaphase 1? And I've been told that age
Before
comes
beauty.
before beauty, so we have to go
You
back
go
to you again.
first, I
got to
guess.
go first. Eh? All right. There we go. They want to know what error took place in anaphase 1. Now, guys, what happens in anaphase? The chromosomes separate and move towards opposite poles. Now, watch this diagram. There's the contraction of the spindle fiber, and wow, there's nothing there, and the whole set moves to one side only. So the whole set goes to one side only. Okay? But they want to know, let's see how they ask the question. What error? What is the name of this error? When this happens, that means the chromosomes did not separate. What do we call it? Non-disjunction. Big word. Junction means to separate in this case. Non-disjunction, non-separation of the. But notice it is the whole set, not just one chromosome or two chromosomes, but the whole set moved to one side only.
Aslam, while you're on that, Simiso asks, what is the difference between aneuploidy and non-disjunction? Simiso, aneuploidy and non-disjunction.
Good. These two words are connected, Katlego.
Yes.
Non-disjunction can cause aneuploidy,
Okay.
and it can cause polyploidy.
Okay.
And as we're going to go through this question, we're going to distinguish between those two words. Those two words are more different: aneuploidy, and polyploidy. But non-disjunction simply means that the chromosomes do not separate.
Okay.
Either one chromosome or two, or the whole set.
Okay.
Okay? Now we move on. So we said that's the story there. Now the question says, name the error that you described in question 3.3.3. Okay? And this error would be polyploidy. Let's go back, because I'm seeing that earlier I said to you they asked to explain. So this answer that we gave you, non-disjunction, we just put it in brackets for now. We would simply say that the chromosomes chromosomes did not separate. Right? Now we go to the next question, and that goes out. They say name. There we would say, here we would say non-disjunction. Notice here they ask you to name it. Sorry about that, I read wrong there. So you must also be very careful when you're reading the question. What will the chromosome number in A be? This is a zygote. That was diploid. The ovum was diploid. The sperm is haploid. So n plus 2n would give you n plus 2n would give you 3n. So it would be triploid. Triploid. Okay? What is this phenomenon in A known as? Because of that, we call it polyploidy. That's where polyploidy comes in. Well, we said we're going to explain the difference. Why do we call this polyploidy and not aneuploidy? Okay, so let's go back to the diagram. The important thing is the whole set of chromosomes did not separate. And when that joins with so it's a diploid now, in other words, it did not become a haploid ovum, it was a diploid ovum, and it fused with a haploid sperm, so it became triploid. And this process is known as, this phenomenon is called polyploidy. Now, going back to the earlier question, where if only one of these chromosomes sets did not separate, or two of them did not separate, not the whole set, then that is known as aneuploidy. And an example of aneuploidy in your syllabus is Down's syndrome. So that is the difference between polyploidy and aneuploidy. In aneuploidy, it's not the whole set. In polyploidy, the whole set does not separate. So one gamete sits with double the number of chromosomes that it should have. Mention any two advantages of this phenomenon named in question 3.3 in plants. How does this help plants? We can get larger fruit. For example, those lovely juicy Katlego strawberries.
Oh, yes.
They are octaploid.
Oh,
That means they are 8n. 8n. Octaploid. We can also get seedless fruit. Like the bananas are seedless, they're also by polyploidy. We can also get more fruit. And we can get hardier fruit. In other words, they would resist all diseases and all the other things that take place in that field as well. What is the significance of this process? Which process? Polyploidy, in the evolution of plants. Now, this question is so interesting because it touches on 3 sections of your work in one go. It deals with evolution, it deals with meiosis, and it deals with chromosomes and goes back to DNA as well. So it's touching, and in other words, almost all of your paper 1 items are merged in one question. And this is the essence of life science, guys. You need to be able to know that there's a connection with everything you learn, especially in paper 1. Everything is connected somehow. And you see, how is this significant? You must remember that every particular organism has a specific, every species has a specific chromosome number. Now, when this plant has changed its chromosome number, then this, whatever plant it was, it cannot be called the same as its own, as the species that it was before, because it has a different chromosome number. So it leads to speciation. Why? Because it has changed the chromosome. If they had to ask you to explain, then you would say that. If you say it leads to speciation, already you're getting your 1 mark. It was just a 1 mark question. But if you had to explain why that happens, because the chromosome number has changed over this period, and therefore it is now a new species. The next question, a little bit longer question again. The table shows the result from an experiment to investigate the effect of selective breeding for protein content in milk of 2 varieties of milk-producing cows. They say start 10, 20, 30, 40, 50, and the different generations, and selected for high protein content, and this is the protein content in milk, and in cow B, selected for low protein content, and that is the protein content. You look what's happening there, and look what is happening there. Supply a hypothesis for the experiment. Let's go back. Experiment to investigate the effect of selective breeding for protein content in milk of 2 varieties of milk-producing cows. So the hypothesis must be linked to your investigation. And your hypothesis must be a statement. So we'll say selective breeding in the first one increases, you could have said increases or decreases the protein content in the cows. Hypothesis, independent variable, selective breeding. What does it, and what in protein content, what does it do relationship, increasing or decreasing? The next question, plot 2 line graphs on the same axis to reflect the results of the experiment. I would have loved to given you time to actually plot this on your own, and then we could compare notes and you could have marked it. But time, unfortunately, waits for no man. All right? So we're going to look at how we would have plotted this graph. And there's your graph there. This is what you have given. Look at the ticks that are given here. Your heading must have both variables. Your y-axis gets a mark for your label. Your x-axis gets a mark for the label. Look at the intervals. You're getting a mark for that. Intervals getting a mark for that. And you have plotted 2 of these line graphs. For giving a key, you're getting a mark. And for your plotting, you're getting marks according to the rubric that is there. Any questions, Katlego?
There's no questions so
There's
far.
no questions so
I think you've
far.
gone through everything.
So guys, this is how you draw your graph. Today's, the skill should be based on learning the graph. So please make sure that you learn that graph. Katlego.
Awesome. Thank you, Aslam. What an awesome show, guys. I'm so glad you guys enjoyed it. Life science is always such a breeze and time goes by so fast. But thank you for joining us today. It's been such a great show, and I hope you guys learned so much more. Remember that on Saturday, there is a show, guys, so don't worry. There's more, okay? And from us, thank you for joining me today, and thank you for all your great comments. Bye-bye, guys.